I will now be discussing with you Streptococcus, and Streptococcus is still part of our gram-positive cocci. Okay, so Streptococcus is one of the gram-positive cocci. So it is part of the gram-positive cocci module, and what you would actually notice here is that the organisms are actually cocci, and they appear in chain-like manner. So they appear in chains. Okay, and through the years, there has been many numerous taxonomical changes to Streptococcus. So by the way, when you say Streptococcus, this pertains to the family. Okay, and there has been many numerous changes because of the molecular analysis due to DNA homology and DNA sequencing studies. So these are molecular analysis. So before, they have this, they usually classify them according to letters. So this particular letter classification is what you call the Landsfield Classification. Before, they have this group known as Group D, but now Group D is classified as Enterococcus. And then Group N is now classified as Lactococcus. So there has been many numerous taxonomical changes. So this is one of the biggest family. Streptococcus E is one of the biggest family because there are at least 17 genera. And of course, the most familiar or the most popular of which would be the Streptococcus. So I did mention to you, excuse me, a while ago the Landsfield Classification. So this was actually the work of Rebecca Landsfield. So she was the scientist who studied Streptococcus at its fullest extent. So Rebecca Landsfield was the one who discovered that at the cell wall of Streptococcus, we have a certain carbohydrate. So we call it the C carbohydrate, polysaccharides. Okay? So the C polysaccharide carbohydrates. So this particular C polysaccharide carbohydrates serves as the main antigenic characteristic. Okay? Ito nga yung Landsfield carbohydrates. The main antigenic characteristic of the Streptococcus. Okay? So what does it do? Um, this particular C carbohydrate polysaccharide in the cell wall of beta hemolytic Streptococcus would be the reason why we can divide Streptococcus into several serologic types. Okay? So serotypes or serologic types. And these particular serotypes would have letter assignment. Okay? So for example, if you are group A, meaning to say you have the same characteristic of C carbohydrates. If you are group B, you have the same character. If a certain organisms belong to group B, they have the same characteristics of C carbohydrates. Okay? So the C carbohydrates, therefore, is the antigenic basis, for classifying serological types of Streptococcus. Okay? So let's discuss the general characteristic of Streptococcus. Okay, so these are the characteristics of Streptococci and Enterococci. So noteworthy to mention that when we say Enterococci, these are cocci that mostly live in our gastrointestinal tract. Okay, so most of them are facultative anaerobic. What does it mean? It means that uh, they are aerobic by nature, but but can live without oxygen. But there are some who behave like aerotolerant anaerobes. So which means that they grow in the presence of oxygen but do not use it for respiration. Okay, so that's one of the behaviors or the atmospheric requirements of Streptococcus. Okay, they grow best on enriched media with blood or serum, primarily because Streptococcus are considered to be as a fastidious organisms. So when we say fastidious organisms, it means that they would require additional nutrients such as enriched medium, okay, in the form of blood. So you would notice that they would grow in sheep's blood agar plate, and they would exhibit different hemolytic patterns, which we will be discussing again later on. But if you have already watched my discussion on Staphylococcus, I have already discussed there the different hemolytic patterns of the organisms. Okay, so there are gram-positive cocci seen in pairs, and some of them are actually in chains, and they are catalase negative. Okay, so remember, Staphylococcus is catalase positive. Streptococcus is catalase negative. Okay? So again, if you remember the principle of catalase test, which I have already discussed in the previous discussion on Staphylococcus. So when you say catalase, we are using hydrogen peroxide or agua oxygenada as the reagent. And the enzyme catalase is supposed to hydrolyze it into water and oxygen. And the presence of oxygen results to bubbles formation or rapid effervescence of gas, which serves as a positive result. Take note that we cannot get samples from blood agar plate because our blood naturally contains catalase enzyme, and it will result to false positive result. So that's the reason why instead of getting samples from blood agar plate, um, we should get it from other medium. Let's talk about the carbon dioxide requirements. Okay. So one of the most important characteristic of streptococci is that they are capnophilic. So when we say capnophilic, it means that these organisms would require increased concentration of carbon dioxide. Okay, so that's one of the simplest way of providing carbon dioxide for Streptococcus is to use a candle jar. So this is an example of a candle jar. So you pile up your petri dishes here, and then you place you place candle on top of it, and then you lit the candle. Now, once you cover the jar, so oxygen will be consumed and eventually the candle will burn out, and the smoke that will come out from the candle will provide CO2. But of course, a more sophisticated and a more accurate way of providing CO2, particularly if you want to achieve 5 to 10% CO2, is to use a CO2 packet. So it's just like CO2 contains in an ordinary pillowcase, and then all you have to do is to open it and put it inside the jar. So it's actually a more sophisticated but a more expensive way of providing carbon dioxide. So growth of other species is increased in the presence of CO2, but it is not required. So colonies of growth are usually small and transparent. So again, guys, do not forget, whenever we say small colonies, particularly whenever we are pertaining to Streptococcus, we are actually referring to pinpoint colonies. Okay, so we are referring to the pinpoint colonies. So these are the appearance this would be the appearance of Streptococcus when stained using a gram stain. So they are actually dark purple or violet. Okay, so some of them are in pairs, some of them are in a very long chain, some of them appears in tetrads, but most importantly, they are in chains. Okay, so that's Streptococcus. So by the way, the left is in the solid medium and the right is in the liquid medium, from the liquid medium. Okay, so if we will be talking about the cell wall of Streptococcus, we would expect that they have a very thick peptidoglycan layer, primarily because this is common, sorry, this is common for gram-positive organisms. So we'd expect them to have a very thick peptidoglycan layer. And teichoic acid is also present for gram-positive organism. Okay, the C carbohydrate layer is present in all groups except for the viridans group. Okay, so that's the reason why the viridans group do not have Landsfield classification. They do not have Landsfield classification because the viridans group do not have C carbohydrate. So again, I did mention to you that C carbohydrate contributes to the antigenic property of Streptococcus. They are encapsulated in some species. So not all Streptococci would have capsule, but one of the species that would have capsule is Streptococcus pneumoniae. Streptococcus pneumoniae, and in young cultures of most species. Okay. Hemolysis, we have already discussed hemolysis in Staphylococcus, which I've already uploaded. Hemolysis is the lysis of RBC. And again, there are actually three main types of hemolysis. Beta hemolysis refers to complete hemolysis. So RBCs here are completely raised. So there would be clear area. So the surface of the blood agar plate becomes colorless because it is a complete hemolysis. When you say alpha hemolysis, it is incomplete hemolysis. Meaning to say, RBCs are merely partially raised, so resulting in the greening of the area around the colony. So greenish brown hemolysis is another way of describing the surface of the blood agar plate. So there would be a greenish brown hemolysis. Alpha prime hemolysis, so there's another one. Alpha prime hemolysis has an intact zone of RBCs with hemolysis around them. Okay? And then lastly, we have the gamma hemolysis. Gamma hemolysis means that there's no hemolysis. And since there's no hemolysis, therefore you wouldn't expect any changes in the color of the agar. So it remains red because there's no hemolysis. Okay, so these are the three types of hemolysis. Okay, so again, you can also review these particular types of hemolysis in your book. So we are in chapter 15 of Maycon. And by the way, alpha prime hemolysis is sometimes described as the wide zone of hemolysis. So again, this is an example of the beta hemolysis. So when you say beta, you'd expect that there's a colorless zone of, this is the colorless zone. Okay, this is the colorless zone. And then this is the colony. And this is supposed to be greenish brown. Supposed to be greenish brown for alpha hemolysis. Okay, so let us now discuss the clinically significant streptococci and streptococci-like organisms. So, general characteristics. So, do you know that Streptococcus is associated with many clinically significant streptococci and streptococcus-like organisms, and there has been known diseases associated with them for over 100 years. Can you just imagine that a simple toothache may lead to heart disease? So that's the reason why they say that a health if you have a healthy teeth, you'll also have a healthy heart. So that's actually true because organisms, for example, streptococci in your in your in your teeth may actually travel down to your heart, causing you to have endocarditis, so things like that. Okay, so these organisms may be characterized as beta hemolytic or pyogenic, or non-beta hemolytic or non-pyogenic. Okay, so that's the classical way of categorizing them. But then later on, because of the research of Rebecca Landsfield, Landsfield classification would be a more systematic way of classifying them, except for the viridans group, because again, as what I mentioned, viridans group do not have the C carbohydrates. Okay, so again, this table is found in your book. So we have the classification such as the group A Streptococcus, representative organisms would be Streptococcus pyogenes, and it is classified as type A, and it can cause beta hemolysis. Group B Streptococcus, representative organism is Streptococcus A Galaxy. It is known as the Group B Streptococcus, and it can cause beta hemolysis. Group C Streptococcus, our representative organisms are Streptococcus dysgalactiae and Streptococcus equi. So this one is type C A. And then group D, which has been reclassified as Enterococcus, which I will be explaining to you later on. So we have several classification as well. And then Streptococcus pneumoniae doesn't have also a Landsfield group, but the common term for that is pneumococcus. Okay, so we will be discussing them in details, including the different diseases that can be caused by these particular organisms. Okay, so let's start our discussion with Group A Streptococcus. Okay, Group A Streptococcus is known as the Landsfield Group A. So sometimes it is classified as the gas group or the Group A Streptococcus. And perhaps one of the most important characteristics of the group A Streptococcus is the presence of M protein, which could serve as one of the virulence factors for group A Streptococcus. Okay, so M protein is attached to the peptidoglycan of the cell wall. So group A Streptococcus, as what I've mentioned to you a while ago, our representative organisms is Streptococcus pyogenes. So the term pyogenes refers to pyogenic, which means that this particular organism is capable of forming pus. Okay? So this organism is not usually considered as part of the normal human flora, as it is the strain of Streptococcus most of the time associated with the disease. So if you are actually suffering from strep throat, kung nagkakaroon kayo ng pharyngitis, one of the most likely culprit would be the Streptococcus pyogenes. Because if you will be looking at the site where we can find them, they are mostly confined at the upper respiratory tract, such as in our nasal vestibule, nasal entrance, nasal cavity, internal nares, soft palate, nasopharynx, epiglottitis, and larynx, and even in our lower respiratory tract. So it can cause both upper respiratory tract infection and lower respiratory tract infection. So let's talk about the virulence factor. Okay, so I did mention to you a while ago the M protein. And M protein is essential for virulence because of the presence of the M protein, a Streptococcus pyogenes can evade phagocytosis. And then there are even 80 different types of M proteins, and they have a number designated to it. For example, M5, M10, okay? And usually we resist infection against Streptococcus pyogenes if we have already developed antibodies related against or antibodies to M protein. Okay, so that's a good news if you have antibodies against M protein. But in case that we do not have, then it's actually a dangerous kind of bacteria. Later on, I will be explaining to you what are the different diseases that can be caused by the group A Streptococcus. Aside from M protein, we also have the protein F. Protein F is short for fibronectin binding protein. And protein F, together with lipoteichoic acid, these two are known as the adhesion molecules. Meaning to say, if this is your epithelial cell, fibronectin is here on top of your epithelial cell, and because of that particular adhesion molecule, Streptococcus can easily adhere to your epithelial cells. See? So that's the reason why it is considered as the adherence factor. Because of the presence of lipoteichoic acid F protein complex, it will now be easier for Streptococcus to attach to your epithelial cells. And then another one is capsule. Capsule, as we all know, can evade phagocytosis. Okay, so capsule can evade phagocytosis. So it allows the bacterium to mask its antigen and remain unrecognized by its host. Okay, so what you can see here is an example of a capsule, a swollen capsule. A swollen capsule is actually known as the Neufeld-Quellang reaction, and it is usually positive for Streptococcus pneumoniae. Okay. So what else? Another type of another type of um, virulence factor would be streptolysin. Do you know that there are two types of streptolysin? So we have streptolysin O, and the other one is streptolysin S. So streptolysin O is responsible for hemolysis on blood agar plate. And since it is oxygen labile, it is responsible for subsurface hemolysis. While streptolysin S is responsible for surface hemolysis, the one that is exposed, okay, because streptolysin S is oxygen stable. Streptolysin O is oxygen labile, meaning to say it is only active in the absence of oxygen, which means it is only active under anaerobic conditions. So streptolysin O can destroy WBC, platelet, RBCs, and other tissues. The same thing with streptolysin S. But streptolysin S can do it even in the presence of oxygen. The thing is, streptolysin O is antigenic. Streptolysin S is non-antigenic. And because streptolysin O is antigenic, we can use this as a serological test to check if we have been recently exposed to a streptococcal infection. We call this ASO test, anti-streptolysin O. Okay? So anti-streptolysin O is actually a serological test for streptococcus. And if you are positive with ASO, it means that you have been recently exposed to streptococcal infection. So we will be talking about more of this in next sem if you will pass bacteriology. Of course, you will pass bacteriology. Um, there's a subject next sem called serology and immunology, and wherein ASO will be discussed. But just so to give you a background, the normal range of ASO is less than 200 international unit per ml. Meaning to say, if you are more than, if you get a result of at least 200 or more than 200, it means that you are positive. And if you are positive, it means that you have a streptococcal and post-streptococcal infections. Okay. Other virulence factors for group A would also include the DNases, and there are four types of them: DNases A, B, C, and D. And DNases will help destroy foreign DNA. And the most common type of DNase would be the type B. And then hyaluronidase is the spreading factor. So this, we have already discussed this one in Staphylococcus. Hyaluronidase can destroy the matrix, the 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 matrix or the ground substance in the connective tissue. So once it has been broken down, then it can now the infection will easily spread. And then we also have the STEs. Okay, so streptococcal pyrogenic exotoxin, formerly known as the erythrogenic toxin, and it is the reason why we have rashes. We have rashes, okay, in scarlet fever. So SPEs are actually can act as super antigen, and there are actually several types of them, such as the type A, type B, type C, and type F SPEs. Now, there are several tests that we can actually do, okay, to determine if we're positive with scarlet fever. So one of them is Dick's test. Dick's test is a skin test, so do not get me wrong. So we inject erythrogenic toxin into the arm of the patient, and if there are rashes, it means that the patient is positive for scarlet fever. And then another one is the Schultz Charlton phenomenon, or the blanching phenomenon. So here we inject anti-erythrogenic toxin. Here in Dick's test, we inject erythrogenic toxin. So Dick's test will tell us if a person is could most likely be positive for scarlet fever. Schultz-Charlton phenomenon will tell us, okay, if a certain person with rashes, and it will tell us if those rashes are actually caused by Streptococcus pyogenes. So if we inject anti-erythrogenic toxin and the rashes disappear, then that rash is caused by Streptococcus pyogenes. Okay, if we inject anti-erythrogenic toxin and the rash didn't disappear, means that it is not caused by Streptococcus pyogenes. So that's the reason why it's called the blanching phenomenon or the Schultz-Charlton test. Okay? So let's talk about the infections or the diseases that can be caused by Streptococcus pyogenes. And one of them is perhaps one of the most common, the bacterial pharyngitis and tonsillitis. And nowadays, it is quite alarming to have it because you might think that you might have COVID, but then not all tonsillitis is caused by COVID. It can be bacterial by nature. Okay, so This one is also known as strep throat and common between ages of 5 to 15 years. So it has an incubation period of 1 to 4 days. So you'll have sore throat, malaise or body weakness, fever and headache. So the disease would vary in intensity and symptoms, and usually it will subside in 3 to 5 days unless there would be complications. And it's very important that we treat it with antibiotics. Why? Because if not treated and if we allow it to persist, there's a chance of us developing post-streptococcal infections, which I will be discussing later on. So, what are these post-streptococcal infections? Do you know that post-streptococcal infections are actually post-streptococcal infections are actually the consequences. So we call it the sequelae, and it's actually more dangerous than the actual pharyngitis itself. So there's also a chance for us to develop skin or pyodermal infection through bites or abrasion to the skin. So usually we have impetigo. This is an example of impetigo. Okay, a localized skin disease, usually in very young children. And then erysipelas. So this is erysipelas. Skin infection with spreading red rash with demarcated but irregular edge. Demarcated irregular edge, mostly in older adults. And cellulitis. It is the deep invasion, as you can see here, of group A streptococcus leading to necrosis and gangrene. So that's the reason why in some cases, group A streptococcus is sometimes known as the flesh-eating bacteria. So they are also known as the flesh-eating bacteria. And then of course sepsis is also equally dangerous because when you say sepsis, it means that the organisms are already found in blood, and if they are found in blood, it means that they are multiplying also. So a while ago, I did mention about scarlet fever. Okay, so scarlet fever is also known as the strawberry tongue or the pink tongue. Okay, so it is caused by SPEs, which I've already discussed, and it is carried by a T12 bacteriophage, meaning to say, due to um, lysogenic bacteriophage transduction. Okay, so di ba, we discussed that already. So when say transduction, the genes are being injected by lysogenic bacteriophage. So what will happen here is that rashes appears on the chest and spread to the trunk, limbs that would last for about 5 to 7 days. So after the rashes, skin will desquamate. Okay, so as you can see, it's characteristic of strawberry tongue or pink tongue is one of the um, classical signs of having a scarlet fever. And another one is the necrotizing fasciitis. So another reason why they call it flesh eating, a flesh eating disease. So infection is characterized by rapidly progressing inflammation and necrosis of the skin, subcutaneous fat, and fascia. So, necrotizing fasciitis is life-threatening, and it is characterized by type 1, type 2, and type 3. And among these three types, the one that can be caused by group A streptococcus is the group 2 or the type 2. And then there is also what you call streptococcus pyogenes or streptococcal toxic shock syndrome. And another dangerous disease because the entire organ system will be affected. Okay, it will lead to death. So it is initiated by severe streptococcal infection. So at first, patient would have bacteremia, and then they will have necrotizing fasciitis. So at-risk populations are those very young children, especially with chickenpox, and also older adults. So it may be a result of a complication of chickenpox, because if children have chickenpox, their skin, sometimes they are actually scratching it, and then their skin is exposed, and an exposed skin can actually lead to secondary bacterial infection, which could be Streptococcus pyogenes. Okay, so moving forward, a while ago I told you about you have to you have to take a simple strep throat seriously, because if not, it may lead to post-streptococcal infections. Okay. Do you know that post-streptococcal infections are actually more dangerous than the actual sore throat itself? So the consequences are actually more dangerous. And one of them is called rheumatic fever. So rheumatic fever will start after pharyngitis, and it will result into inflammation of the joint, heart, blood vessels, and subcutaneous tissues. And one organ that we do not want to get affected here is our heart, because once there is a serious damage in the heart valve, it will now result to rheumatic heart disease, and eventually uh, serious complications will happen, which can be fatal. So there could be cardiac arrest. Okay? So that's dangerous. Rheumatic fever or rheumatic heart disease. And another equally dangerous one is when when the organism, okay, goes to your kidneys, and it will now cause acute glomerulonephritis. So again, it can occur after pharyngitis or even after cutaneous infection or skin infection. So it is actually an immunologic mechanism resulting into the antigen antibody complex, or also known as the immune complex. Why? If, for example, this is the basement membrane of your kidneys, and then there would be immune complex. So immune complex means that antibodies, okay, this is the antigen, and then this is the antibodies, and then this is the complement. So we call it immune complex. So immune complex will be deposited at the basement membrane of your kidneys. And once it happens, your own WBC will attack the immune complex, damaging the kidneys as well. And if renal failure occurs, a person could be in constant dialysis, or worse, if there is a total renal failure, this can lead to death of the person. So that is the reason why it is very important not to ignore a simple sore throat, because it could probably be a strep throat, and a strep throat can lead to serious complications. So how do we diagnose? First of all, you have to be familiar with proper specimen collection. So when I say proper specimen collection, so we use swab and we rub it over the posterior pharynx at each tonsillar area. So there are two tonsillar area, and we have to rub it left and right. Okay? If exudate is present, okay? If exudate is present, it should also be touched with the swab. What is an exudate? So exudate is actually a liquid or white patches, and it is actually a result of inflammatory process. Okay? So we should be able to touch it. We should avoid contamination by tongue and uvula, and then we place it in sheep's blood agar. Remember the organism, at a while ago I told you in the introduction that the organism is fastidious, and it would require enriched medium. And then we strict it in blood agar plate, and one of the things that we seriously miss here in online class is the actual streaking in blood agar plate. So we can streak it and we call it multiple interrupted. Okay, ideally, the blood agar plate should contain sulfometoxazole. Why? The presence of sulfometoxazole serves as the inhibitory substance. It serves as the inhibitory substance. It improves the recovery of beta hemolytic streptococci from the throat culture after 24 hours. So if there's no growth, then you have to wait for another 24 hours. Okay, so sometimes when we when we do the throat swab and look for the blood agar plate, one of the things that we have to look into is the presence of hemolysis. Because if it's a greenish brown, meaning to say the organisms are alpha hemolytic, you might have just isolated normal flora and they are not clinically significant. So and one of the things that you have to confirm in order to know or in order to check if you have isolated Streptococcus pyogenes is to look for the presence of beta hemolysis. So when you say beta hemolysis, there is a complete form of hemolysis. So these are the traditional tests that we are doing for Streptococcus pyogenes. So one of them is the bacitracin test. And in bacitracin test, we are using a 0.04 unit of taxo A. So that is the reason why you can find letter A here. So if there is a zone of inhibition, it's positive for taxo A. And then PYR test, so Streptococcus pyogenes would have a cherry red color and Landsfield typing. So you'll have to use anti-sera. Okay? So other tests which I will be discussing at the end of the chapter would be the Kipurate hydrolysis, CAMP test, Val-Escalene, sodium chloride test. I will be discussing that later on. Okay, but these are traditional tests. Take note that traditional tests um, will determine the phenotypic characteristics. But if you really want to know the exact accurate genotypic characteristic, then we have to do the PCR. Okay? So rapid detection tests would also include enzyme immunoassay and optical immunoassay. Okay, so commercial system is already available for direct identification. So these are actually available from various suppliers. So we have the Verogen gram-positive blood culture nucleic acid test, the film array blood culture, and nowadays one of the most popular tests for the exact identification of the organisms would be the Malditoff test, the matrix-assisted laser desorption ionization time of flight bacterial identification system. So why is it very nice? Because first of all, um, it doesn't require you to have it doesn't require you to culture. You can directly get from the clinical specimen. It will give you the result of the organisms and at the same time allow you to perform antibiotic susceptibility testing. Okay, so not all hospitals have Malditoff. So one of the hospitals in the Philippines that would have it, this includes San Lazaro, St. Luke's, and Malditoff test. Okay, so this table is very important. So please study this table. This table will tell us how to differentiate biochemically, phenotypically organisms, particularly Streptococcus pyogenes. So for Pyogenes, it is positive for PYR, and LAP, leucine aminopeptidase. For Streptococcus A Galaxy, it is positive for hippurate, CAMP, and LAP. Okay, so our next topic will be Streptococcus A Galaxy. So that's the reason why I'm actually introducing that to you. And speaking of Streptococcus agalactiae, this particular organism is known as the GBS. So Streptococcus pyogenes is gas. So this time we are now talking about the GBS. So it possesses the group B specific antigen, which is acid stable polysaccharide. So do you know that there are 9 recognized capsular polysaccharide serotypes, and they are designated as 1A, 1B, and type 2. Okay? So they have capsule. And one of the most important characteristics of the GBS is, take note of this one, the presence of sialic acid capsule. And it is weakly immunogenic and can inhibit activation of alternative complement pathway. Okay, so where can we find Group B, hemolytic streptococcus? So they are found as part of the normal flora of the female genital tract. So these are the areas where we can find them. Okay? The female genital tract. Normal flora. It's normal for female, but it is not normal for neonates. Okay. Meaning to say, newborn infants, okay, who would be passing through the birth canal, would have chances of getting Streptococcus agalactiae. And it may result to neonatal meningitis, neonatal sepsis, and even death of the infants. So let's talk about the GBS virulence factor. So one of them is capsule. So we all know for a fact that capsule prevents phagocytosis, but it is ineffective after opsonization. So sialic acid, which I have mentioned now for the second time, is the most significant component of the capsule and critical virulence determinant. Okay? In some mutant strains, they cannot help it, but there are some Streptococcus agalactiae that would lose the sialic acid. So if sialic acid is lost, there would also be loss of virulence, which is fortunate on our part. But if the Streptococcus agalactiae would have sialic acid, this will make them more virulent, more dangerous. So this is how they look like under the electron micrograph. So products that are being produced are hemolysin, the CAM factor, DNases, hyaluronidase, and proteases. I think I haven't discussed protease here. Protease here is actually an enzyme that can actually destroy protein, particularly the immunoglobulin, which is the antibody, basically our antibody. And then the CAMP factor. CAMP is actually an acronym which stands for Christie, Atsky, Munch, Peterson. And what is the characteristic of the CAMP factor? The characteristic of the CAMP factor is this. I hope you can see this. Okay. We call it the arrowhead hemolysis. I hope that when I do lecture, you have your notes with you, because most of the time, I'm mentioning things that are not found here in the PowerPoint presentation. Okay? So, therefore, if the organism is positive with CAMP, Christie, Atsky, Munch, Peterson, is characterized by arrowhead hemolysis. So later, what is this one basically? So you have here blood agar plate. So this is a blood agar plate. And then you have here, so if this is the blood agar plate, you place the gas here. Place the gas here. And then you place S aureus in the middle. And then you place GBS on the other side. Notice that between the gas and S aureus, nothing happens. But between GBS and S aureus, there is some form of an arrow. So this is the characteristic of the camp factor, which is actually quite unique for Streptococcus A Galaxy. So a while ago, I told you that Streptococcus A Galaxy is normally found in the female genitourinary tract. They're normally found in female genitourinary tract. But that is exactly where neonates will pass through during delivery. So, in some cases, these poor neonates will get Streptococcus agalactiae, and there would be significant cause of invasive disease, leading to death of infants. But in United States, according to them, cases of these neonatal deaths have dramatically decreased since 2008, primarily because they now have GBS screening as part of maternity care or prenatal care. So, paano ba yung GBS screening? So, later on, I'll be explaining what do you mean by GBS screening. So, there are actually two types of infections with GBS. So, the first one is the early, and the other one is the late onset infection. So, when we say early onset infection, it means that it occurs in less than 7 days. And 80% of cases is actually due to vertical transmission. Again, when we say vertical transmission, it is from mother to infant during birth. So, early onset may result to premature birth, membrane rupture, and of course, um, patients or neonates could develop pneumonia and meningitis, and it results in a very high mortality if not treated quickly. Okay. The late onset occurs for neonates 7 days and older, usually between 1 week to 3 months of age, and it is primarily characterized by meningitis. So we do not want any of the early or late onset infections. That is the reason why a proper prenatal care is very important. Okay. Now, how about for adult infection? Okay. So, adult infection, so mother after childbirth or abortion can actually suffer from endometritis and endocarditis. And also for older adults, particularly if you are immunodeficient, due to the skin and soft tissue infections, intra-abdominal abscess, bacteremia, and pneumonia. So again, it is normal in genitourinary tract, but not in the endometrium and definitely not in the heart. So if there's an infection in endometrium, so we call it the endometritis, and endocarditis is at the inner wall of the heart muscle. So the treatment for Streptococcus agalactiae would include ampicillin with or without an aminoglycoside. A while ago, I was telling you that prenatal care is very important, which means that um, suspected pregnant women should be screened by having the vaginal and rectal swab between 35 and 37 weeks of gestation. Just to be sure, just so to check if they have GBS in that particular site, particularly if they will have normal delivery. Okay? So, usually the doctors would be collecting vaginal and rectal material and inoculate it in the following culture media, such as the Todd Hewitt broth with antimicrobials, or another choice would be the trans vag broth or the strep B carrot broth. So, what are these? The broth serves as the enrichment medium. And after the enrichment medium, for example, becomes turbid, then we subculture it to sheep's blood agar. So the sheep's blood agar is the enriched medium. So usually if it's broth, we have to subculture it in the primary medium. And in this case, the primary medium is the sheep's blood agar plate, which serves as the enriched medium. Since Streptococcus is fastidious. So how they would look like in blood agar plate. So in blood agar plate, SGBS appears like grayish white mucoid colonies surrounded by small zone of beta hemolysis. So this is the beta hemolysis, and then this is the grayish white mucoid colonies 24 to 48 hours after and incubated at 35 degrees centigrade. So if you will be looking them under the microscope, they appear like gram-positive cocci in chains. So shorter chain in clinical specimen, but longer chains if the organisms would be coming from the culture. So the presumptive identification would be based on biochemical reaction, and of course, the most common is the CAMP test. So this is how we differentiate GBS from gas. So of course, they are catalase negative. Kasi kapag catalase positive, that is Staphylococcus. So we perform bacitracin this test. So taxo A. If it's susceptible, definitely that is a gas. But if it's resistant, it could be GPS. So we do the CAMP test. If it's positive, it is GPS. But if it's negative with CAMP, then possibly group D or beta hemolytic strep, not in A, B, or D. Okay? So this is now our algorithm in differentiating gas from GBS. So citracine is the key using a 0.04 unit of taxo A. So how do we know if it's susceptible? There is what you call zone of inhibition. So if there's a zone of inhibition, it means that it is susceptible. So if you notice, hippurate hydrolysis is another test for Streptococcus A Galaxy. So this is a positive result, the presence of a purple color. And the arrowhead hemolysis. Arrowhead hemolysis is a positive reaction for GBS. So the Center for Disease Control of the United States recommends universal, which includes the Philippines, vaginal and rectal screening of all pregnant females in late the third trimester. So screen pregnant women with asymptomatic bacteriuria, meaning if during urinalysis they found out that there are many bacteria in the urine, then we have to screen them for GBS. So prophylactic antimicrobial therapy for women who sample test positives with vaginal and rectal screening, urine culture, women with previous history, and unknown status of GBS at the time of delivery. So it's actually being recommended so that infants will not die from neonatal meningitis or neonatal sepsis. Okay, so let's talk about the group C and G streptococci. Do you know that the group C and group G streptococci are divided whether they have large colony isolates or small colony isolates? So when we say large colony isolates, this includes Streptococcus A Galaxy, subspecies Equisimilis. Sometimes it is classified as type A or L. So primarily because they are beta hemolytic. So again, paano natin nalaman na beta? As you can see, colorless structure here, the surface of blood agar plate. And then the other one is Streptococcus aeruginosus. Sorry. I'm sorry for that. And okay, so let's talk about another group of Streptococcus. But this time, let's talk about Streptococcus pneumoniae. Okay. Streptococcus pneumoniae, um, the nickname is pneumococci. Okay. The Streptococcus pneumoniae contains C substance as well, similar to carbohydrates of other Landsfield group. But take note that there is no Landsfield classification for Streptococcus pneumoniae. And Streptococcus pneumoniae would also have capsular antigen with approximately 19 known types. And the capsule is antigenic and can be identified with appropriate anti-sera. So again, if you will be looking at the cell wall, so there are many virulence factors found primarily in Streptococcus pneumoniae, such as the pneumolysine, neuraminidase, autolysine, so on. But one of the most important characteristic of the capsule is that it became positive. It could become positive in anti-capsular serum known as the Neufeld-Quelling test. So again, I have introduced that to you a while ago. And Neufeld-Quelling test is an example of capsular swelling, where the capsule swells upon treatment or upon mixing with anti-capsular serum. So another example of a swollen capsule. So the virulence factors of Streptococcus pneumoniae will include capsule and then hemolysin, IgA protease, neuraminidase, and hyaluronidase. So what are the infections? Streptococcus pneumoniae is a common normal flora in upper respiratory tract pathogen. Okay? Sorry. It's a common normal flora found in the upper respiratory tract, and at the same time, it is a pathogen. How could that be? If it's a pathogen, meaning to say, it is prevalent in older adults or with other disease, meaning to say, they could serve as a secondary infection. For example, for someone with COVID-19, COVID-19 is a viral infection, right? But since you are already immunodeficient, if you have COVID-19, there's a chance for you to get secondary bacterial infections in the lungs as well. And that particular bacteria could be Streptococcus pneumoniae. So it says here, after viral infections. That's the reason why COVID patients are also being treated with antibiotics to prevent secondary bacterial infections. Okay? So infection begins with aspirations of the organisms. So that's the reason why um, there's also a vaccine against Streptococcus pneumoniae, and it's very important that we get these vaccines as well. Okay? Otherwise, once we get infected with Streptococcus pneumoniae, the type of pneumonia is known as the lobar pneumonia. So lobar pneumonia is characterized by chills, cough, dyspnea, or shortness of breath. So it leads to the edema of the lungs. So there's a feeling that it is as if you're drowning. Mortality rate with 5 to 10%, and without therapy, it jumps close to 50% mortality. So Streptococcus pneumoniae can result also to sinusitis, otitis media, both from children under 3 years old, and then bacteremia. When you say, well, the three otitis media is the infection of the middle ear. Luga in Tagalog. Meron kayo makita ng ganyan expression. And then, bacteremia. Okay, so bacteremia is the presence of bacteria in the blood, but it will actually pass through the blood-brain barrier system. Will pass through the blood-brain barrier system, and it will pass through the blood-brain barrier system. So what will happen is that meningitis will actually go. Kasi yung blood-brain barrier system, meaning to say, the bacteria will go to the CNS. Then meningitis could happen. So both follow the previous infections, creating spread or over the wide area of the body. So this becomes invasive. Bacteremia and meningitis is invasive, and this could be, this could be fatal as well. Okay. Streptococcus pneumoniae clinical infections can also be involved in endocarditis. Again, this is the inflammation of the heart muscle. And peritonitis, um, all from dissemination. So, this is our peritoneal cavity, kung nasan yung bilbil natin. And that could also be infected. And it could also result to hemolytic uremic syndrome after you have been positive with blood and CSF specimen. And usually, a person will be falsely positive with Coombs test. Coombs test is the direct antiglobulin test. Antiglobulin test in blood banking. This could actually complicate cross-matching results, but in the blood bank. So, hemolytic uremic syndrome means that usually hemolytic uremic syndrome is caused by E. coli 0157H7, particular strains of E. coli that can cause hemolytic uremic syndrome. But Streptococcus pneumoniae may also affect our kidneys. That's the reason why hemolytic uremic syndrome is listed herein. So, lab diagnosis, what you can actually see in the gram stain are gram-positive diplococci. When you say diplococci, they are in pairs. And they are lancet shaped. So, lanceolate. Sometimes that's how they describe it. As culture ages, gram stain reaction becomes variable. And then, there would also be capsule stain. Ang ibig sabihin ng becomes variable. Sometimes, kapag sobrang tagal na ng culture, and then you stain it with gram stain, you might get gram-negative results. And this is a false negative. Capsule stain can reveal the capsule. So, usually capsule is characterized by halo surrounding the organisms. So, there would be halo surrounding the organisms. So, Streptococcus pneumoniae, okay. So, the culture media and colony appearance appears like this one. So, complex media required brain heart infusion, SBA, and chocolate agar with increased CO2. Again, kapag sinabing increased CO2, it means that they are capnophilic because they require carbon dioxide for growth. So typically, there is a large zone of alpha hemolysis on blood agar surrounding the colonies. So, how would you know if it's alpha hemolysis? If this is the blood agar plate, you would notice that there is a greenish brown hemolysis. Okay? So, young culture, usually, there is also a greenish brown, but you would notice that the colonies are round, glistening wet mucoid dome-shaped appearance. Yan. So, ito yung nag-glisten. So, usually kapag nag-age, yun yung makikita ninyo. Nag-greenish brown and all. So, ito yung toxopy. Toxopy is short for optogene test. Optogene test is the screening test for Streptococcus pneumoniae. Okay, so usually, meron siyang zone of inhibition. Meaning to say, Streptococcus pneumoniae is susceptible to toxopy or optogene test. So as the colonies becomes much older, autolytic changes will result in collapse of each colony, giving it like a coin-like raised rim. So ito, medyo old na. So kapag napansin nyo, kapag old na yung colonies, nag-autolysis, ibig sabihin, namamatay na yung mga bacteria. And if that happens, um, that happens, uh, wala na yung glistening factor. Hindi katulad nito, ito yung mga young culture, meron kayong makitang glistening factor. So again, diplococci, lancet shape, and then greenish-brown hemolysis, glistening colonies for Streptococcus pneumoniae, and this is supposed to be small letter P. And another small letter P. Okay, so identification methods. So the key traditional test would be the bile solubility test. So we place the organisms in bile. So Streptococcus mitis is not lysed by bile, it's our negative control. Streptococcus pneumoniae is lysed by bile, so there is a clearing. And then optogene susceptibility test, the toxopy, there is a zone of inhibition. So by the way, the active component of toxopy is ethylene hydrocyprine. So the active component is ethylene hydrocyprine for toxopy. And then other available tests would include the letter P, Fadebach pneumococcus test. You know what this Fadebach pneumococcus test? Fadebach is actually a good test for CSF because it will not just test pneumococcus, it can also test for Streptococcus B, for Neisseria meningitis, and for Haemophilus influenzae. So it's commercially available. Malditoff, which I have already explained to you. And then there's even a Streptococcus pneumoniae rapid antigen test using urine sample. And for a blood culture identification panel, film RA-BCID is also available in sophisticated laboratory for identification, rapid identification of Streptococcus pneumoniae. So, Streptococcus pneumoniae is generally treated with penicillin, but the problem with penicillin, many antibiotics have become resistant to it already. So that's the reason why erythromycin or chloramphenicol can be used as an alternative for bacteria Streptococcus pneumoniae that have become resistant with penicillin. Vaccine is very important, especially nowadays. Okay, vaccine against most common capsular antigens. So it is recommended for following, no? splenic individuals, older adults, and cardiac patients. These are people with comorbidities. It helps reduce incidence and severity, particularly nowadays. So after receiving COVID-19 vaccines, perhaps you can also receive, after several months of interval, the Prenevar. So this is commercially available for Streptococcus pneumoniae, and even the flu vax. It's very important so that we prevent secondary bacterial or viral infections. Or even my baby has already received pneumococcal vaccine. So can we differentiate pneumococci from other streptococcus? Actually, yes. So again, we did mention of biosolubility and optogene test and Neufeld-Quelling. So aside from biosolubility, Neufeld-Quelling, and optogene test, we also have inulin fermentation. So Streptococcus pneumoniae is an inulin fermenter. They're also susceptible to quinidine and positive for mouse virulent test, wherein the mouse dies within 16 to 48 hours upon injection with Streptococcus pneumoniae, while other Streptococcus will not immediately cause the death of the mouse. Okay, so this is how we differentiate pneumococci from other Streptococcus. And now let's discuss the viridans group. Okay, so when we say viridans group, again, they are normal flora of the upper respiratory tract, female genital tract, and even the gastrointestinal tract. Many of them would show alpha hemolysis, so characterized by greenish brown hemolysis. They are fastidious and would require carbon dioxide, so they are capnophilic. And the viridans have five groups. We have the mitis, mutans, salivarius, bovis, and anginosus. Noteworthy to mention is Streptococcus mutans. This is an example of anaerobic organisms. Anaerobic organisms and Streptococcus mutans would be responsible for dental caries. So the uh, virulence factor, so these are ito nga, yung mga noteworthy to mention, are able to form biofilm on the tooth surface. Ito yung tinatawag natin dental plaque. So, if for example, after we have eaten and we cannot immediately clear the food debris, Streptococcus mutans, in the absence of oxygen, will ferment the carbohydrates, and one of the products of carbohydrate is acid. Acid metabolites are important in the formation of dental caries. Why? This is the enamel of our teeth, of our tooth. So the acid being produced can actually destroy the enamel until such time that the dental pulp is reached. The dental pulp is where the root or the nerve is found. So once the enamel is destroyed because of the acid production by Streptococcus mutans, this will now reach the nerve, and upon reaching the nerve, this is the time that we can feel something. We can feel something as part. The tooth is already aching. Yan. So that's where we can feel something. Okay? So the virulent streptococci, the virulence factors are not well established. So they could have the polysaccharide capsule and cytolysine, and these have been identified in some member, particularly of the anginosus group. Other factors with possible virulence tendencies, such as the extracellular dextran, self-surface associated proteins, and extracellular enzymes. Perhaps one of the really clinically significant would be Streptococcus mutans, as it is already established as the main etiologic agent of dental caries. But do you know that the virulence group are generally can cause opportunistic infection? So again, when we say opportunistic infection, it means that they take advantage of hosts with weak immune system. since they are part of our oropharyngeal commensals. Meaning to say, if after performing throat swab and we find greenish brown hemolysis, that cannot be significant because it could be part of the normal flora. But for people whose immune system is weak, that is significant because virulence group may actually lead to opportunistic infection. And also, virulence streptococci is one of the most common causes of SBEs. So when you say SBEs, we are referring to the subacute bacterial endocarditis. More common in children than in adults with hematologic malignancies. The heart valves are damaged by rheumatic fever. Okay, yung may mga gas, they are prone to have SBEs as well. Gas means group A streptococcus. Okay, so other diseases would include tooth decay, meningitis, abscess, osteomyelitis, and empyema. So they say if you have unhealthy tooth, you are also at risk of getting heart disease because the organisms can actually go to your heart and cause SBEs or subacute bacterial endocarditis. Fortunately, majority of these virulent streptococci are susceptible to penicillin. So, yan. So, this is another example of a damaged heart valve due to the virulent streptococci. So, how do we diagnose virulent streptococci? So, they are extremely difficult to identify isolates. So, laboratories should be satisfied to place isolates into any one of the 5 groups, muties, mutants. Okay. Culture colonies are small and surrounded by a zone of alpha hemolysis. Although some strain may either be beta hemolytic or non-hemolytic. So really it's difficult to identify virulent streptococci. And one of the things that we can also do to screen is because of the fact that all viridans are pyrrolidose negative, but they are lab positive. So there's a special kind of disc known as the leucine aminopeptidase, and when we place the culture, there would be pinkish changing color of the disc, and that's positive for lab test. So all of the viridans group are positive with the lab test, leucine aminopeptidase. So what are the 5 groups again? The anginosus, the S. bovis, the S. mitis, the S. mutans, and the S. salivarius group. So nowadays, there are now newer means of identifying Streptococcus viridans. So this includes the development of molecular methods such as the DNA hybridization technique and PCR. The multi-locus sequence analysis may be used to identify unknown streptococci, and of course, MALDI-TOF system are already in the evaluation stage. Okay, so let's talk about the viridans streptococci group B versus the versus the enterococci. So this is where changes have actually happened. Okay, so Streptococcus bovis group, ito yon, Streptococcus bovis group. Okay, and enterococci has a common denominator. So S. bovis and enterococci have common denominator, and that is both of them possess the antigen. Therefore, Streptococcus bovis is no longer a valid species name recently. So this has been published 2019, no? So DNA studies found that S. bovis and S. equinus were the same species. Okay? Both group both groups are bile-asculin positive, but non-enterococci were 6.5% sodium chloride negative, whereas the enterococcus were 6.5 sodium chloride positive. What is 6.5 sodium chloride? 6.5% sodium chloride is what you call the salt tolerance test. This will test whether the organisms can tolerate high salt concentration, such as the presence of 6.5% sodium chloride. Okay? So both enterococci and non-enterococci group, okay, both of them can lyse or clear the bile esculin, as shown here. Okay? But the non-enterococci cannot tolerate 6.5% sodium chloride, but the enterococci group can tolerate 6.5% sodium chloride. Therefore, the enterococcal group is now the genus Enterococcus, and the non-enterococcal group is part of the D Streptococci. I hope you're getting it, okay? So, enterococcal, before kasi, we have the enterococcal and non-enterococcal. Enterococcal group is no longer called enterococcal group. Ang tawag na sa kanila is Enterococcus. And non-enterococcal group is now not known as enterococcal group, but they are now known as the GDS, group D Streptococci. Okay, so Enterococcus is different from the GDS. And how do we differentiate them? Ability to grow at 6.5% sodium chloride. Negative. Enterococcus group is 6.5% sodium chloride. Positive. So the group D sometimes manifests endocarditis, UTI, and wounds. Okay? So one of the ultimate way of distinguishing group D from Enterococcus is the Enterococcus is resistant to penicillin, while group D is susceptible to penicillin. So again, we no longer have the classifications of enterococcal and non-enterococcal group, but instead we have the Enterococcus and the GDS. So enterococcal is now known as Enterococcus, non-enterococcal group is now known as the GDS. So again, this is how GDS look like on blood agar plate. Enterococcus look like in blood agar plate. So greenish brown hemolysis indicating that they are alpha hemolytic. So to sum up what I have discussed, okay, if there's no hemolysis on blood agar, we do the bile esculin. Sorry, this one pala, wala palang hemolysis ang enterococcus. Stand corrected on that matter. So if there's no hemolysis on blood agar, we do the bile esculin. If it's positive, we do the 6.5 sodium chloride test. If it's negative, we classify them as the non-hemolytic streptococci. If it's positive with 6.5 sodium chloride, we call it the Enterococcus group. If it's negative, we call it the group D streptococci. And how do we differentiate alpha hemolytic streptococci from enterococci? Okay. Alpha hemolysis on ship's blood agar plate. Again, let me stand corrected now. It's still possible for enterococcus to be alpha hemolytic. As shown here, there is a greenish brown hemolysis. Okay. So if there's alpha hemolysis on ship's blood agar plate, we should be able to differentiate it from enterococci. So we do the optogene disc. Remember the optogene? It is the taxopi. If there is a zone of inhibition, it means that it is susceptible, therefore it is Streptococcus pneumoniae. If it's resistant, we do the bile esculin. If it's negative, then viridans. But we do the salt tolerance test and the PYR. Positive positive means it is Enterococcus. Negative negative, it is group B Streptococcus. So these are the two algorithm. If we have the first algorithm that we discussed is differentiating it from non-hemolytic Streptococcus. The second algorithm is differentiating it from alpha hemolytic Streptococcus. So, the general characteristic of Enterococcus, okay, so why do we call it Enterococcus? The term entero refers to gastrointestinal tract because it is a natural inhabitant of both human and animal intestinal tract. So, the most common species are Enterococcus faecalis and Enterococcus faecium. So all species possess the D antigen. Thus, they belong to the group D antigen, GDS. So it will result into weak bubbling in catalase test. Remember, remember, Streptococcus are supposed to be negative in catalase test. But for Enterococcus, we call it the pseudo-catalase because of the weak bubbles. It can grow under extreme condition like bile salt or alkaline pH. So, grow in extreme condition, resistant to multiple antimicrobial agents. They also have the surface adhesion proteins. They have extracellular serine protease, which can destroy our IgA. They can have the enzyme gelatinase, which obviously can lyse gelatin. And they have two subunit toxin called cytolysine. So this is the toxin found in Enterococcus faecalis. So Enterococcus would most likely cause nosocomial infection. So when we say nosocomial infection, we are referring to hospital-acquired infection. So we are referring to hospital-acquired infection, and UTI is the most common, oftenly associated with urinary catheterization or other urologic manipulations. So immediately, prolonged usage of catheter can result to UTI, prolonged hospitalization, and cons- and complications can include bacteremia and endocarditis. In fact, Enterococcus endocarditis accounts for 5 to 10% of bacterial endocarditis. So, again, I've been telling you this. So, there are, this is another way, this is, I've been presenting again the slide to tell you how to differentiate Enterococcus from the rest of the group D by 6.5 sodium chloride so they can tolerate salt. So, characteristic of Enterococcus, okay, there should be standard collection and handling procedure for the isolation of the specimen. Specimen should be cultured immediately, and we can culture the specimen in TSB, tryptic soy broth, or BHI agar, supplemented with 5% sheep's blood. And organisms grow well at 35 degrees centigrade in the presence of CO2. So again, the organism is said to be capnophilic. So, Enterococcus can be weakly positive in catalase test. This is not. This is a strong bubble in catalase test. This is the negative result. So somewhere in between is the weak bubble. So unfortunately, I cannot find an image of a weak bubble. So that is for Enterococcus. And again, they can grow in extreme condition. Kaya na they can tolerate salt, they can lyse bile, and they can even grow an alkaline pH. So these are the different species of Enterococcus. So aside from Fecalis and Faecium, we also have the Durans, Avium, Cassilliflavus, Gallinarium, and Raffinosus. And they can be differentiated according to different tests such as the motility, fermentation with mannitol, sorbitol, arabinose, and raffinose, and telluride medium, arginine, and then the PYU or the pyruvate, and then the MGP or the methyl alpha D glucopyranoside. Okay, so these are the different ways of phenotypically differentiating different species of Enterococcus. And unfortunately, Enterococcus have already acquired resistance to many aminoglycosides, to beta-lactams and glycopeptides. And there's even a report of VRE, vancomycin-resistant enterococci. And 8 phenotypes have already been described, but the most common phenotype are the Van A and the Van B. So usually, we use vancomycin-containing agar and chromogenic VRE medium in order to screen. So if the organisms cannot grow in vancomycin-containing agar, so ibig sabihin, that organism have already developed vancomycin resistant. And to be more accurate with your test, then of course, you can always use PCR. Okay, so we have come to the second to the last part of our lecture. So bear with me. We are actually discussing 126 slides in here. So second to the last topic would include the clinically significant streptococci and the SLO. So we call it the streptococcus-like organisms. So four genera in this group may resemble the Virulence Streptococcus. They are isolated in clinical specimen and they are associated with infections resembling streptococci and enterococci. So they are not really streptococci. So we call them the SLO or the streptococci-like organisms. This includes Aerococcus. So you can see the morphology here. Appears like Streptococcus, Gemella, Lactococcus, Euconostoc, and Pediococcus. So these are the most common SLOs. So aside from that, we also have the Abiotrophia and let me pronounce it again, Granulicatella and Abiotrophia. Okay? This particular bacteria grows as satellite colonies. So Abiotrophia grows with Granulicatella side by side. And we consider them as the NVS or the nutritionally variant streptococci because they would require additional compound in the medium for them to grow, such as the sulfhydryl compounds or the presence of 10 milligrams per liter of pyridoxal hydrochloride. So these are the things that we are adding in the culture medium for the NVS to grow. And they can cause bacteremia, endocarditis, and otitis media. And they are difficult to treat with antibiotics. So let's talk about the SLOs one by one. So we have here the Aerococcus. So incidentally, Aerococcus is a common airborne organism. So it is an opportunistic pathogens that may cause bacteremia, endocarditis, and UTIs. So they resemble virulent streptococci in the culture primarily because of the alpha hemolysis. So they are gram-positive. They are also pseudo-catalase positive, and susceptible patterns would be similar to enterococci. So medyo nakakalito sa laboratory. And then we also have the gemella. Okay? So colony morphology, also similar resemblance to the viridans. They are either alpha hemolytic or non-hemolytic, and they can easily decolorize during gram staining. Kaya napagkakamalan siyang gram-negative cocci. So sometimes they appear in tetrads, pairs, or in clusters, or in short chains. And then we also have here the lactococcus, which may appear singly in pairs or in chains, physiologically similar to enterococci, and they are either hemolytic. Hello? Sorry for that. Okay, so they are either alpha hemolytic or non-hemolytic, and they are previously classified as the group N streptococci. So this one is associated with UTI and endocarditis. Leuconostoc, gram-positive, and they are irregularly coccoid morphology. So you can actually the irregular morphology here under the electron microscope. So they share the same phenotype and biochemical characteristic with Lactobacillus, Viridans, Pedococcus, and Enterococcus. They are also associated with bacteremia, UTI, meningitis, pulmonary infection, and intrinsically resistant as well to vancomycin. No? Dumadami na talaga yung resistant sa vancomycin. Okay? So this is the gram stain of the conostoc. So take note of the irregular coccoid colony. And the colonies of leuconostoc here as how they appear in blood agar plate. Very small pinpoint colonies, non-hemolytic patterns or gamma hemolytic patterns. Pediococcus, which could appear in tetrads, as you can see here, by pairs or in cluster. Resembles streptococci or enterococci. So pediococcus can cause bacteremia, abscess, and meningitis. Resistant as well to vancomycin and associated with infections. Particularly, those patients who have underlying GI abnormalities or previously had abdominal surgery. So this one is linked to bacteremia, abscess formation, and meningitis. And the last part of today's lecture, we will just recall the laboratory diagnosis that we have discussed since there are several of them. So again, hemolytic patterns, um, we can classify organisms according to hemolysis. Actually, we call it the Smith and Brown classification. So when we classify them uh, according to hemolytic pattern, call it the Smith and Brown classification. When we classify them according to physiologic characteristics, we call it the Burgess classification. And when we classify them according to the surface typing, we call it the Landsfield classification because we classify it according to the serotyping, capsular polysaccharide, and surface protein. And then, of course, biochemical characteristics. So, again, you should be familiar already by now with hemolytic patterns. So, we have the alpha hemolysis, incomplete hemolysis, characterized by greenish brown. Beta hemolysis, characterized by colorless colonies, and gamma hemolysis, which means that there's no hemolysis. But there's the fourth type, which is the alpha prime or the wide zone. So the alpha prime or the wide zone is characterized by small areas of intact RBCs around colonies surrounded by a wider zone of complete hemolysis. So that's called alpha prime hemolysis. And then the physiologic characteristics. Okay. So when you say physiologic characteristic, this is the academic or Burgess classification. So we have the pyogenic group. So again, when you say pyogenic group, they are fast-forming organisms and constitute most of the Landsfield classification. Do you know that we can also classify them according to growth at 45 degrees centigrade or growth at 10 degrees centigrade? So usually, pyogenic group cannot grow at 45 and 10 degrees centigrade. Okay? And then, we also have the lactococci. So sometimes it is known as the lactic group, such as yung Streptococcus lacticola. Do you know that the lactic group cannot grow at 45 degrees, but they can grow at 10 degrees? And then we have the Enterococcus group, which is part sometimes of the normal intestinal biota. So the Enterococcus group can grow at 45 degrees and 10 degrees centigrade. And then the very dense group, such as yung mga strep mutans, strep salivarius, okay, it can grow at 45 but not at 10 degrees centigrade. And so this is the physiologic characteristics of, so this is another way of classifying Streptococcus according to the physiologic characteristics. And syempre, one of the most important um, classification is the Landsfield Classification, developed by Rebecca Landsfield as early as the 1930s. Okay, so what she did is that she placed the streptococci in a diluted acid solution and heat it for 10 minutes. So that becomes a soluble antigen. So that particular solution, what she did is she injected that into a rabbit to immunize the rabbit. So naturally, the rabbit will develop antibodies to the antigen. And then that particular antibodies are now being used to differentiate the different antigens. So currently, there are designated as A, B, C, D, F, and G. So there are actually six, but if you notice, no, we just discussed GAS, GBS, Group C, very short, Group D, very short lang yung discussion natin. And then we didn't even have a glimpse of discussion on Group F and Group G because some of them are actually animal pathogens. So another way is biochemical identification. So this is the key test to determine presumptive identification, such as the hemolytic pattern. So it is exhaustive, ibig sabihin nakakapagod, and it is not always needed. Okay? So such as this one, this is the algorithm, okay, on how to differentiate um, Staphylococcus from Streptococcus. I will not be discussing it one by one because technically I have discussed some of them already in the previous slides, but this is not the overall picture. So you may want to check your book. It's actually found in your book, or can screenshot it and study it later on. So there is a rapid identification test that we do not have to do it in the tube. So we usually do it in um, rapid ID system, which is commercially available. So they're being guided by color code, and it will be positive or negative. And this will actually lead you for the presumptive identification of Streptococcus. So we discussed a while ago the bacitracin susceptibility testing using the Taxo-A disc, which is consisting of 0.04 unit. Zone of inhibition means it's positive. No zone of inhibition is resistant, therefore it is negative. We can also use PYR disc, and the reaction is more definitive. CAMP test is an acronym for Christy, hindi siya Kampa, so let me correct this one. Christy Atkins Munch Peterson. So these are the scientists who developed the CAMP test. So there are three ways of doing it. You can use the beta lysine producing SRUs. So this is the SRUs. SRUs produces beta lysine. Or you can use a disc that is impregnated with beta lysine, and more recently they have developed the rapid CAMP test. So how do we describe it? It's described for as arrowhead hemolysis. Usually, CAMP test is the screening test for group B streptococcus. Screening test for group B streptococcus. So the principle is that it detects the production of enhanced hemolysis. So the enhanced hemolysis is the reason why there is an arrowhead hemolysis. It occurs when beta-lysine, hemolysin, the source of beta-hemolysin is S. aureus, and the group B streptococcus, this is the group B streptococcus, come into contact. So if S. aureus and the group B streptococcus come into contact, there would be an enhanced hemolysis. So we can also use purified beta-lysine on confluent strep colonies, and these that may contain beta-lysine would also work. So this is the rapid CAMP test. So this is also known as the SPOT test. And then we also have the heparate hydrolysis test, which could differentiate group B streptococcus from other beta hemolytic strep. So the principle is that the group B streptococci possess the enzyme hypuricase. So hypuricase is short for hypurate hydrolase, which can hydrolyze sodium hypurate, forming sodium benzoate and glycine. So after you have added ninhydrin reagent, so sodium benzoate and glycine plus ninhydrin reagent will react, forming a purple complex, and that is a positive result for hypurate hydrolysis. And then the PYR test, or the complete name of the PYR is the pyrrolidonyl alpha naphthylamide hydrolysis, or PYR test, is a presumptive test for group A streptococcus and non-group D streptococcus. So gas is the only streptococcus species that is PYR positive, and other positive genera are the Enterococcus, Erococcus, and Gemella. So a positive reaction is presence of a cherry red color, or you can also do the PYR disc, simply streak it with the colonies, and if it becomes pink, if the disc becomes pink, then that's also a positive result. So the PYR test detects the activity of substrate L-pyrrolidonyl arylamidase, so also known as pyrrolidonyl aminopeptidase, which is being hydrolyzed by group A Streptococcus and Enterococcus. It is also as specific as 6.5% sodium chloride for Enterococcus, and more specific than bacitracin for gas. So between bacitracin test and PYR test is more specific, meaning to say, chances of getting false positive result would become very minimal if you're using PYR test instead of the bacitracin test. So this is another way of doing the PYR test using a disc. Leucine aminopeptidase, or the LAP test. Okay, so we have the LAP disc wherein we smear the organism, incubate for 5 minutes, and then there would be hydrolysis of leucine beta naphthylamide, and the release of beta naphthylamide will become red after you have added the DMACA reagent. The DMACA reagent stands for para-dimethyl aminocinnamaldehyde. Okay? So it is also useful in differentiating along with the PYR test. So Aerococcus and Leuconostoc would usually be negative. And then the Vod's-Proskauer test. It's another biochemical test for Streptococcus. It detects acetone produced from glucose. So in a modified Vod's-Proskauer, so we have to use a heavy suspension of bacteria with 2 ml of Vod's-Proskauer broth, and then we add 5% alpha naphthol and 40% potassium hydroxide. We shake it for 30 minutes at room temperature so that we dissolve oxygen, and if it becomes pink, then it's positive. And then we also have the beta-D glucuronidase test, or the BIGUR, which detects the action of beta-glucuronidase. So what is beta-glucuronidase? It is the enzyme found in the isolates of large colonies forming group C and group G beta-hemolytic streptococcus. So it is not found in small colonies. So kapag large na yung colonies ng group C and group G and they are beta-hemolytic at the same time, that's the only time that we'll be using the BGR test. So it's commercially prepared. Rapid tests are also available. So this one is a positive result. And then the bile esculin test or the bile esculin hydrolysis test, the principle is that sometimes some organisms would have the ability to grow in the presence of 40% bile and at the same time hydrolyzed esculin. And these are important features of organisms such as Streptococci that possess group D antigen. So Enterococci would also be positive with this. And then of course the salt tolerance test. Salt tolerance test or this one will test the ability of the organisms to grow in 6.5% sodium chloride, which will allow us to differentiate group D from enterococci. So usually group D will grow in salt and sorry, enterococci will grow in salt, but group D will not grow in salt. So it is being done alongside with PYR. Kaya yung result na enterococci ay positive, positive. Ang group D ay negative, negative. The optogene susceptibility test or the TAXO-P. Okay, it contains ethylene hydroxyprene hydrochloride. So that this is added on the surface of the sheep's blood agar, inoculated with a suspected alpha hemolytic strep. We incubate it for overnight and measure the zone of inhibition. If the zone of inhibition is greater than 14 millimeter, if you're using the 6 millimeter disc, or if it's greater than 16 millimeter if you're using the 10 millimeter disc, that is considered as a positive result, which means that the organism is susceptible to optogene, and if the organism is susceptible to optogene, then you're able to detect presumptively Streptococcus pneumoniae. Here, there is no zone of inhibition. Here, it is positive for zone of inhibition. TAXO-P, optogene based. And the bile solubility also, it's different from bile esculin. So, it takes the advantage of S-pneumoniae to autocatalytic enzyme amidase. So, under the influence of bile salt or detergent, the cell wall lyses during cell division. So, it's diagnostic also for Streptococcus pneumoniae. Nagkaroon ng clearing. And of course, we also have the non-cultural identification, meaning to say you don't have to culture. We detect directly from the clinical specimen, such as um, immunoassays using direct clinical specimens, such as a throat swab, or we isolate the colonies, extract the C carbohydrates, and use it agglutination test. We can do the latex, slide, or enzyme. So this is an example of the slide agglutination test from group A to G. And a more expensive test would be the nucleic acid probes using the real-time PCR and then the DNA probe, wherein we should be sequencing the 16S rRNA, which is, of course, sequences that we're using for prokaryotes. And for susceptibility testing, take note that penicillin is the drug of choice, but of course, resistant is not a remote possibility. So in cases of resistant, alternative choices would include erythromycin and narrow-spectrum cephalosporins. So multi-drug resistant have also been reported for Streptococcus pneumoniae and vancomycin-resistant enterococci. So select combination of drugs that can be effective based on susceptibility patterns and the body sites of specimen origin. Yun, so thank you very much for listening.